Hello, welcome to our channel. This year's Nobel Prize in Physiology or Medicine goes to Mary Branco, Fred Ramstel, and Shimon Sakaguchi for their groundbreaking discoveries that revealed how our immune system achieves what scientists call peripheral immune tolerance. Their work has provided the world with a fundamental understanding of how our immune defenses stay active against invading microbes, yet remain restrained enough to avoid attacking our own cells. It's also a discovery that could transform how we treat autoimmune diseases and even reshape the future of immune-based therapies. As we know, our immune system has T cells to help us fight infection, and without them we cannot live long. However, some T cell receptors will unavoidably recognize proteins and structures in our own bodies, so-called self-reactivity. These self-reactive T cells may become harmful and cause autoimmune diseases such as type 1 diabetes, multiple sclerosis, and rheumatoid arthritis. So, how does our body solve this problem? From early life, our T cells are generated in the bone marrow and then travel to the thymus where they go through a test. In this test, harmful self-reactive T cells are eliminated in a process called central tolerance. For a long time, this was believed to be the only way self-tolerance is obtained. However, some self-reactive cells escape out into our circulation and are potentially dangerous. This year's Nobel Prize in Physiology or Medicine concerns the discovery of how those potentially harmful T cells are kept in check. The discovery started with Shimon Sakaguchi's interest in the phenomenon that removing the thymus from mice 3 days after birth led to an overreactive immune system which caused autoimmune diseases. However, if T cells from healthy mice were injected, the development of autoimmune disease was prevented. Sakaguchi realized that among these injected cells, there must be some cells that protect against the overreactive immune system and autoimmune diseases. He started a long search for these cells. Years later, he discovered that if the T cells to be transferred were depleted of cells with the cell surface protein CD25, this pool of cells would no longer protect against autoimmune diseases. But if the T cells with CD25 were also injected, the mice were protected. Sakaguchi called these CD25 positive cells regulatory T cells. Many researchers were, however, skeptical about the existence of regulatory T cells. The critical missing piece would come from Mary Branco and Fred Ramstel. They studied a mouse strain called scurfy because of its scaly skin. The scaly skin was part of the severe autoimmune disease the mice developed, and other researchers had shown that the scurfy mutation was somewhere in the middle of the X chromosome. To find the mutation causing the disease, Branco and Ramstel established that the X chromosome section of interest contained 20 potential genes. They then examined gene after gene. It was only with the 20th and last examined gene that they found the mutation, and they named the new gene FOXP3. Branco and Ramstel suspected that the rare autoimmune disease IPX, which is also linked to the X chromosome, could be the human variant of the scurfy mouse disease. To understand if that was the case, they analyzed samples from children with IPX and found mutations in the FOXP3 gene. These findings connected the FOXP3 gene also to human autoimmune disease. These two key discoveries, Sakaguchi's identification of cells that regulate immune responses and Branco's and Ramstel's identification of the FOXP3 gene as important for keeping the immune system in control, indicated that the FOXP3 gene could be important for regulatory T cells. Shimon Sakaguchi was the first to show that FOXP3 is actually essential for the development of the regulatory T cells, so-called Tregs, and therefore critical in peripheral immune tolerance. This was soon followed by reports from other scientists and unleashed a whole new field in immunology. These insights have also sparked intense clinical interest. Scientists and biotech companies around the world are now racing to harness the power of Tregs for therapeutic use. The goal is to calm overactive immune responses in conditions like autoimmune disease, or to prevent the immune system from rejecting transplanted organs. Currently, no Treg cell therapies have received FDA approval, but several are advancing through clinical development with investigational new drug (IND) clearances and designations like Fast Track status to expedite progress toward potential approval. One leading example is ABA-101, developed by Abata Therapeutics for progressive forms of multiple sclerosis, both primary progressive and non-active secondary progressive MS. This therapy uses a patient's own Tregs, engineered with a specialized receptor that targets damaged myelin, the protective sheath around nerve fibers. In preclinical studies, ABA-101 showed promising results, reducing inflammation and supporting tissue repair in animal models. The FDA cleared its investigational new drug application in July 2024, paving the way for a Phase 1 clinical trial expected to begin dosing in late 2024 or early 2025. In August that same year, it received Fast Track designation to speed development, underscoring the urgent need for new treatments in progressive MS. Another contender is TRX-103, developed by TR1X. Unlike autologous therapies, TRX-103 is an allogeneic, or off-the-shelf, Treg therapy designed to mimic the behavior of natural type 1 regulatory cells. Derived from healthy donors, TRX-103 aims to suppress inflammation and reset immune balance in patients with treatment-refractory Crohn's disease. It received FDA clearance in August 2024 for a Phase 1 trial focused on safety and clinical activity in moderate to severe Crohn's disease. A third major effort is GNTI-122 by GentiBio, targeting newly diagnosed type 1 diabetes. This autologous engineered Treg therapy aims to preserve the body's insulin-producing beta cells by re-educating the immune system toward tolerance. GNTI-122 is built from a patient's own CD4+ T cells, reprogrammed for enhanced stability and specificity to islet antigens. Preclinical data revealed potent suppression of autoreactive T cells, suggesting the therapy could potentially halt disease progression. The FDA accepted its IND in July 2025, authorizing a Phase 1 multicenter trial to assess safety, tolerability, and biomarkers such as C-peptide levels over a 78-week follow-up. As we look ahead, the discovery of regulatory T cells reminds us that the immune system is not just about attack, it's about balance. The science behind Tregs is rewriting how we understand disease, and how we might one day prevent or even reverse autoimmunity. The next few years will test whether these pioneering therapies can move from the lab to the clinic, and from promise to practice. For patients and clinicians alike, the message is clear: the frontier of immune regulation is here, and it's rapidly advancing. What we must pay attention to now is how these therapies evolve, how they perform in real patients, and how this delicate art of immune balance could one day become the foundation for a new era of precision medicine. If you found this content helpful, don't forget to give a thumbs up, subscribe, and hit the notification bell. Thanks for watching, and we'll see you next time.